So today we're going to focus on chapter 21, which will encompass the lymphatic system and immunity. So we know that the immune system is a complex collection of cells and organs, and it destroys or neutralizes the pathogens that would otherwise cause us disease. So the lymphatic system, in other words, is a system of vessels, cells, organs that carries fluids to the bloodstream. Really, it's the excess fluids that it's carrying, and then it filters these pathogens from the blood. So when we look at global health issues from the past and even currently, we know that something like HIV and AIDS, they tend to continue to be major public health issues. We know that this virus targets the immune system, right? And it weakens people's defense against many infections. And then we know that the most advanced stage of HIV is AIDS, also known as acquired immunodeficiency syndrome. AIDS can take many years to develop if it's not treated. And just to bring the science piece in, you can see in that small picture here under letter B, that if we look at this micrograph, we can see the green particles that are budding off of the surface of the macrophagy, which is the pink structure. Okay, functional anatomy of the lymphatic system. Let's look at some of the key functions of the lymphatics. This system is responsible for draining excess interstitial fluid, which we know is the fluid in the gaps from the interstitial space, and transports it to the bloodstream. Once this tissue fluid enters the lymphatics, it is no longer called interstitial fluid. We now call it lymph. And when we have blockage in the normal drainage of lymph, we call it lymphedema. And we know that edema is swelling. The lymphatic system houses the phagocytic cells and the lymphocytes that clean the tissue fluid before it's dumped into circulation. It also absorbs the digested fats from the intestine. So those are really the key functions. And let's look a little bit more closely at the anatomy. So we have the lymph vessels, which are going to essentially carry the lymph from the peripheral tissues to the venous system, right, to the veins. We have underneath that category the lymph capillaries, which we know are present in almost every tissue and organ in the body. They're actually believed to be as abundant as blood capillaries. And then naturally, these capillaries, although they are throughout the body, they are lacking in the areas of the body. Actually, they're absent in the areas of the body that lack a blood supply. So when we think of some examples, we think of the cornea of the eye, and we think of, the central nervous system, the bone marrow. These capillaries are lined by simple squamous epithelium, which we learned about in A&P1. Then also as part of the lymphatic structure, we have the lymph collecting vessels as well. We have lymph trunks, and those trunks include the jugular, the subclavian, the bronchomediastinal, and the lumbar. So basically what are trunks? They're lymph collecting vessels that converge to form larger vessels. And basically they're named after the body areas that they drain. And then finally, as far as the vessels go, we have what we call the lymph ducts. And those ducts are basically the lymph trunks merging to form the two largest lymphatic vessels. And specifically, those are the right lymphatic duct and what we call the thoracic duct. And the thoracic duct ascends along the left side of the vertebral column. So you'll just want to be familiar with, generally speaking, the vessels and how they work as far as the lymph vessels go, and then the breakdowns that we looked at with the capillaries, the collecting vessels, the trunks, and once again, the ducts. And this gives us another close-up to look at those tissue cells that we were discussing in more detail. So as far as the drainage goes, you'll want to know that the thoracic duct drains a much larger portion of the body, as you can see here, than does the right lymphatic duct. And once again, to identify those ducts, remember, we're going by the patient's side. So we're looking at the patient's right in this area here in purple, that's the right lymphatic duct drainage. And then on the patient's left in the gold or yellow, you can see how that covers a much larger part of the body. Okay, what happens when there's disruptions in drainage? Well, we talked a little earlier about lymphedema, and you can see that here, where there's a blockage of those vessels that leads to the fluid staying there, causing the swelling. And then I'm not sure if anybody's heard of this, but this is actually extremely rare. And the name of this is lymphatic filariasis, which is also known as elephantiasis. And basically, this is an infection, again, an extremely rare infection that cannot be cured. Basically, what's happening here is, as you can see, it's causing a lot of edema, a lot of swelling in the right leg here. And so basically, this person was infected with a parasite that is a form of a roundworm. And again, once again, this is a very rare thing. Can't be cured, but can be managed with antibiotics. So what are the most important things to know for the quiz and the exam on the organization of immune function? Well, you'll want to know that one of the biggest functions is the barrier defense. In the sense of the barrier defense of the immune system, we have our skin offering a level of protection. We also have our mucous membranes that protect us. And what they're going to basically do is they act right away to prevent that pathogenic invasion into our body tissues. So we have something called the innate immune response, which is a rapid, non-specific response, which consists of a variety of specialized cells. And then we have something called the adaptive immune response, which is much slower, yet it's specific and effective, and it involves many types of cells. Now, the thing with the adaptive response is it's primarily controlled by our white blood cells, which we learned that the fancy name for that is leukocytes. And then these specific leukocytes are known as lymphocytes. All right, so we're talking about the adaptive immune response. We're talking about the white blood cells, the leukocytes, known as the lymphocytes. And they're going to regulate our immune response. So if somebody's having something systemic going on and the physician is trying to figure out what it is exactly, they may run a test to determine if the patient has elevated white blood cells. So we're looking at 3 different classes in that case. We're looking about phagocytic cells. And those are the ones that we know ingest the pathogens to essentially destroy them. We're looking at the lymphocytes, and basically those are coordinating the activities, right? And then we have the granular white blood cells, which are helping to mediate the immune response. All right, so if they're elevated, that's a sign of an infection or other pathology going on in the body. So let's take lymphocytes here and let's break that down into the types of lymphocytes that we should be familiar with. So this course is going to cover all of them: the B cells or B lymphocytes, the T cells or T lymphocytes, the plasma cells, and the NK cells. Those are the natural killer cells. So we can see the percentage of white blood cells that those specific lymphocytes should be composed of within our bodies. All right, so these are the percentage that's within normal limits. And then once again, it tells you the primary function. Just like everything we're looking at the physiology or primary function of what these specific types of lymphocytes do. So when we talk about primary lymphoid organs and lymphocyte development, we want to specifically focus on some key things. We want to focus on the red bone marrow, which are the blood cells that are made in the yolk sac of the embryo. And then as the embryo develops, what happens is this function is taken over by the spleen and the lymph nodes, as well as the liver. Later, the bone marrow is going to take over most of the hematopoietic functions, although the final stages of the differentiation of some cells may take place in some other organs. Then we have, as it shows in the caption at the bottom, we talk a little bit about the thymus. And the thymus produces several hormones. So collectively, they're called the thymosins, and they're important to the development of the functional T cells that we talked about. And thus, they help the body to maintain its normal defenses. So when a person's born, the thymus is very, very large. and continues to grow through childhood as the child is exposed to infection. And then once they reach puberty, the thymus weighs about 40 grams. And then after puberty, what happens is it gradually diminishes in size and it becomes increasingly fibrous. So we call that process involution. And by the time a person reaches 50 years old, the thymus weighs less than 12 grams. So the progression is at puberty, it reaches its max, 40 grams. And then when a person reaches about 50 years old, the thymus is going to be at a weight of about 12 grams. And so there is a direct correlation. A lot of the science shows with the reduction of that thymus and the increase in the susceptibility of a person to infection and disease. And then the secondary lymphoid organs are also important because they also have roles in the immune response, specifically the active immune response. We have the lymph nodes, which are the small lymphoid organs. They range in diameter from really small, about 1 millimeter to about 25 millimeters. We have our lymph node flow, which is going to start with the afferent lymphatic vessels. Those are going to transport the dirty lymph into the lymph node from the peripheral tissue. And then that lymph is delivered to what we call the subcapsular space, which is a big network of reticular fibers and macrophages, and also what we call dendritic cells. Now, these dendritic cells are involved in the initiation of the immune response. And then the lymph from there is going to flow to the outer cortex, which contains the B cells. And then it's going to flow through the lymph sinuses to the deep cortex, which has the T cells. And then it continues into the medullary, the middle sinuses at the core of the lymph node. And this region has both B cells and plasma cells. And then we have, we're talking about the nervous system. We learned about afferent, efferent. Then after the afferent lymphatic vessel response, we have the efferent lymphatic vessel response. And that response is going to drain the clean lymph out of the lymph node, and it's going to exit. Then we also have the spleen. Spleen is the largest lymphoid organ that we have, and it performs the same functions for the blood that the lymph nodes perform for lymph. So what's the spleen's role exactly as part of, being a secondary lymph organ or lymphoid organ? It's going to remove the abnormal blood cells. It's going to store iron, and it's going to initiate the immune response by the B cells and the T cells to the antigens in our bloodstream. And then finally, we have lymph nodules. So what are lymph nodules? Well, they're known as tonsils, right? Tonsils are a large lymphoid nodule, and they're in the walls of our pharynx. So we actually have multiple tonsils. We have palatine tonsils, we have pharyngeal tonsils, we have lingual tonsils, and we have what we call tubule tonsils. And then another type of lymph nodule is called MOLT. And MOLT stands for mucosa-associated lymphoid tissue. It basically protects the rest of the epithelium of the digestive, respiratory, urinary, and reproductive tracts from the pathogens, all right, so from different toxins. So the MOLT components include the appendix, in the sense that if there is an infection or inflammation of this MOLT, what's going to happen is the potential for something like appendicitis, right, which would be the inflammation of the lymphoid tissue around our appendix. We have something called aggregated lymphoid nodules, also known as Pryor's patch. And those are patches. Pryor's patches are found clustered in deep epithelial linings and mainly located in the distal small intestine. Okay, some additional pictures of tonsils and of MOLT. And there's the appendix. So earlier when I was giving you an overview of the immune system, lymphatic system, I was talking about the two different types of responses. So we have the innate immune response and we have the adaptive immune response. So I think this slide does a really good job of breaking down the differences, some of the key features of the two. So what makes a response innate? Well, if it's an innate defense, it's present and functioning beginning at birth. It's non-specific. In other words, they don't distinguish one threat from another threat. and they respond in the same way regardless of the invading agent. These innate responses or defenses tend to be more localized. And so generally they attack the infection where the invading agent is attempting to gain entry into the body. And the innate defenses or responses don't have any specific memory. Regardless of the number of times our body encounters the invading agent, the innate defenses don't improve the response to the infection. They just respond the same way. So innate immunity, in a sense, definitely involves physical barriers. It involves cellular defenses. And so when we talk about defenses, we're talking about phagocytes, and we're talking about the natural killer cells, and chemical defenses, the complement, the inflammatory chemicals. So specifically, we need to know the physical barriers that are helping to keep these hazardous organisms and materials outside of our body. So what are the barrier defenses exactly of this innate immune response? Well, certainly our skin. We learned about the skin. We were talking about the integumentary system. It's the major physical barrier to our external environment. We have the skin being composed of stratified squamous epithelium. We have the keratinized cells, the network of dermosomes that are going to lock other cells together to create that physical barrier. We have the hairs that are found on most areas of our body surface that are going to provide a little bit of protection against mechanical abrasions, especially on our scalp. And they also can help prevent hazardous materials or even insects from contacting our skin. So these epidermal surfaces of our skin are going to receive the secretion of sweat glands, also something we talked about in AMP1. And so basically, these secretions are flushing the surfaces to wash away any microorganisms, any bad stuff. And then the surfaces also receive secretions from the sebaceous glands. So we learned about sebum, which not only lubricates the skin, but it also reduces the amount of free water on the surface of the skin. And it creates a certain environment where most microorganisms would find to be inhospitable. Then we have the mucous membranes, as you can see up here on the slide. Those are the epithelial linings of the digestive, respiratory, urinary, and reproductive tracts that provide us with a barrier that, once again, most organisms cannot cross. All right, under that umbrella, we have the mucus, which is going to bathe the surfaces of our mucous membranes. And basically that mucus is going to capture most microorganisms and debris, and it won't be able to go any farther. It can't gain entry past them, past those membranes. The mucous membranes are also going to secrete chemicals that are going to thereby reduce the growth of microorganisms. And remember, the mucous membranes are held together by tight junctions, and so that also helps create the physical barrier. And then we have the cilia on the mucous membranes that's going to create an outward wave of movement that transports the microorganisms up and out of the body. Now, this mucous membrane tissue or lining possesses the MALT that we talked about, the mucosa-associated lymphatic tissue. And so we talked about the physical barriers there, but what about the defenses if something does enter into our bodies? So if any hazardous organism gains entry into the body, what starts to happen is the cells of our cellular defenses attack the infection at the site of its entry. So what cells exactly are we talking about? Well, similar to what we mentioned earlier, we have the phagocytic cells. Those are the cells that engulf the pathogens and any debris in the cells, should it make it past one of those physical barriers, such as the skin or the mucous membranes. Specific types of phagocytic cells are the neutrophils, the eosinophils, and the monocytes. Then we have the NK cells, which are going to detect and destroy abnormal body cells or any virus-infected cells. So if the body's cells are normal, the immune system actually ignores them. NK cells are also able to recognize the bacteria, the foreign cells, or the cells that are just infected by the virus. And what happens is, in each case, the steps leading to the destruction of the target cell are similar. So infection can also disrupt other internal body systems as well, right? And so infection and other pathologies we talked about can disrupt homeostasis. So we have things pop up like inflammation and fever. Here's another look at the phagocytic cells that we talked about. Once again, the eosinophils, neutrophils, and monocytes are the different types. Once again, the NK cells that detect and destroy the abnormal body cells or the virus cells that are found. So in class, I'll be talking to you more about the process of inflammation as there is movement toward the site of a tissue injury or a wound. And we'll look at the wound healing process in a little more detail than we did in AMP1, bringing in some of these new immune system concepts. So now that we looked at innate immunity in some detail, let's now take a detailed look at adaptive immunity. First, what are the characteristics of adaptive immunity? Well, adaptive defenses, unlike the innate defenses, are not present and functioning at birth. They're instead developed as a result of exposure to various infections. These adaptive defenses are specific, so they're going to recognize and attack one infection with specific antigen while they ignore all other infections with different antigens. These defenses tend to be more systemic, so they can attack the infection anywhere and everywhere in the body, not just where it got in. The adaptive defenses have memory. In other words, with each exposure to the infection, these defenses modify and improve their response to the infection so that the response becomes faster and stronger and longer lasting. These defenses, we can also say, are versatile because the exposure to the infection causes the adaptive cells to multiply. And what happens is clones that are all specific to the infection are produced, and then each clone takes on a different role in the immune response to the infection. And finally, the adaptive defenses exhibit tolerance. So the immune system ignores the normal body tissues, as we said, while it targets the abnormal body tissues. So you can think of like normal body tissues are things that belong to ourselves, right? And then the abnormal body tissues don't belong to us. So those are the ones that the adaptive defenses have been trained to attack. So the two main adaptive defenses that we're going to start looking at in this course are the cell-mediated immunity and the antibody-mediated immunity. So first, the cell-mediated. So what are the properties? Well, these provide the action of T cells, which are produced in the red bone marrow, but then mature in the thymus gland. These T lymphocytes cannot recognize antigen directly. Instead, what happens is the antigen is processed and then presented to the T lymphocyte, and then the antigen presentation occurs, and then when the macrophages patrolling the body tissues encounter the foreign organisms, they engulf them by phagocytosis. And then once the organism's inside the macrophagy, what happens is the macrophagy processes the antigen and exposes it on the surface of its own cell in combination with certain type of protein. And then what happens is specific T cells will destroy the sick body cell by activation of genes within the body cell, which triggers a certain process which disrupts the cell's metabolism. So if we really wanted to get technical, we can break this down even further into various types of T cells. We have helper T cells, we have cytotoxic T cells, suppressor T cells, and memory T cells. But you don't need to memorize each of those for the test. All right, just the big picture things. Okay, if you want to look at this in a little more detail with the specific proteins, you can. And there's the types of T cells that we just mentioned. And also included in the adaptive immune response, we can't forget about the B cells and the antibodies. So we talked about cell-mediated immunity. Now we need to talk about the antibody-mediated immunity. So this type of immunity is going to involve action of the B cells, which are produced in the red bone marrow, and they remain there to mature. We also call this humoral immunity because the B cells attack the infection or their antigens while they're in the body fluids, such as the blood or the lymph that we discussed, or the interstitial fluid. And although these B cells reside in the spleen and the lymph nodes, it's important to know that many are also circulating in the body fluids where they can directly recognize the infection or their antigens, and then they undergo a certain type of expansion. And looking at this from the molecular and cellular level, each antibody molecule is going to consist of 4 polypeptide chains. And then each of the 4 chains is a constant region, and the amino acid sequence is the same. And the variable region where the amino acid sequence is unique. That's going to help form the antibody structure. So you're going to need to be familiar with the humoral immunity, which can be actively or passively developed. So if it's an active development, basically a person makes their own antibodies, giving them long-term immunity. If it's passive, the antibodies are generally if it's passive, the antibodies are generated by someone or something else, which provides short-term immunity. Okay, so here's a closer look at that. So let's look at some examples. So for active, we have, as you can see by the flow chart, naturally acquired active immunity, and we have artificially acquired active immunity. So if it's naturally acquired, a person makes their own antibodies in response to a natural exposure to antigens in the environment. So an example of an environmental impact that would be under naturally acquired would be if one student, let's say, got chicken pox from another student in class. And then for the artificially acquired active immunity, that develops after administration of an antigen to purposely expose somebody to that antigen. So the best example of that would be a vaccination, right? A series of vaccinations. Sometimes it's multiple doses, sometimes one dose. And the goal is to purposely expose somebody to the antigen. So the administration of the antigen stimulates the immune system and right away makes antibodies against that infection, which is why if you get vaccinated for something, you have a less of a chance of acquiring that specific infection. And then we have the passive humoral immunity. So once again, we have same terms, naturally acquired passive immunity, artificially acquired passive immunity. So if it's naturally acquired passive, a person receives the antibodies from another person by participating in some sort of activity. So I guess an example of this would be if an infant was getting antibodies from their mother through suckling on breast milk. All right, that would be a good example. And then for artificially acquired passive, that's where the antibodies produced by someone else are purposely given to a person to provide immunity. So if we're using in the hospital setting or in some sort of first aid setting, anti-venom. So anti-venom is used to treat certain venomous bites and stings. And really, it's really strong. So it's recommended only if there's a significant toxicity or a high risk of toxicity in the body. In the United States, there's an approved anti-venom for rattlesnakes, copperheads, and so forth, certain types of snake bites. And it's based on a purified product that's made in sheep, known as Crofab. And it was approved by the FDA in 2000. And then when anti-venom is not available, what happens is hospitals sometimes will use other types of drugs which delay the effect of the neurotoxic envenomization essentially through the snake bite. All right, so there's other drugs that can be used to delay effects in the body. But the most important part to know is that these antivenoms should be given as soon as possible after the venom has been injected. So now I'm going to kind of talk fast because my goal is to keep every lecture under a half hour. I know you guys have a lot on your plate for AMP2 in a short amount of time. So let's talk about immune system disorders. We've already talked about AIDS in particular. So we know that it's caused by the human immunodeficiency virus, HIV. I want to talk to you about hypersensitivities or allergies. So we can classify those according to different types. Immediate hypersensitivity, also called type 1, would be like your anaphylaxis, for example, where it begins within seconds of exposure. Anaphylaxis needs immediate injection of epinephrine. Something subacute would be considered type 2 or type 3. An example of that would be a transfusion of mismatched blood. I know we talked about that during the cardiovascular system. That would be actually more type 2. And then the delayed hypersensitivity, type 4, would be something like contact dermatitis. So if you were to rub up against poison ivy, for example, that usually is a delayed onset, which occurs within 1 to 3 days of contact. And some more examples of autoimmune diseases where B cells make antibodies against the normal body tissues. And so we have misguided antibodies, which can cause things like MS, multiple sclerosis, or type 1 diabetes. And that concludes my lecture for chapter 21. Let me know if you have any questions.